Let's get the webinar started. Yeah, so first, I'd just like to thank everybody for taking time out to join today's Pixelgen Technologies webinar. My name is Gerald Aguila. I am the Business Development Manager for Pixelgen based out of San Diego, California. First, I'd like to send a thank you to Becca Martin from the Seattle Children's Research Institute for her efforts in both co-promoting and co-sponsoring this event. So thanks again, Becca, for your efforts. We appreciate that. For those of you who are not familiar with Pixelgen Technologies, we are actually a protein interactomics company based out of Stockholm, Sweden. We recently developed a new technology that we will be discussing today. It was released earlier this year called the Proximity Network Assay. This assay has actually enabled researchers to analyze the dynamics of the immune cell surface protein interactome in a manner that has previously not been possible. This has led to a lot of researchers learning new novel insights into disease mechanisms, drug mode of action response, and also biomarker discovery, just to name a few. Today's talk is actually going to be provided by our CEO and founder, Simon Fredriksson, who is joining us from Stockholm today, or tonight for Simon. So Simon actually has over 20 years of experience in this field, and Simon was also the co-founder of the proteomics company Olink. So today's talk, as you can see on the screen today, is entitled Protein Interactomics by Proximity Networks of Single Cells. Simon will give us some deeper details into the proximity network assay, and he will also share some use cases and applications of the technology that we have with some of our collaborators. So before I hand this over to Simon, I just want to go over a couple housekeeping items. First, the session will be recorded, so just want to notify all the attendees that this session is recorded. And secondly, for those who are in attendance during the presentation, please feel free to type any questions you may have during the presentation in the chat box. We will have a question and answer portion towards the end of the talk that we will respond to those questions. If we're unable to get to your question due to time constraints, we will follow up with an email. All right, so once again, thanks everyone for joining today's webinar, and I will hand this over to Simon. So Simon, thank you.
Thank you, Gerald. Thank you for the kind introduction. So very happy to be here this evening and present to you guys our new technology and products around studies of protein interactomics. So we believe that this is the true mechanism, the molecular mechanism behind most cell functions, and we want to enable researchers to study this in translational basic and drug development research. So Pixelgen was started about 5 years ago in Stockholm, Sweden, with life science entrepreneurs and innovators from Olink Proteomics, Halogenomics, Vonadis Diagnostics, 10x Genomics, and Atlas Antibodies, combining expertises from single cell work, proteomics work, advanced nucleic acid amplification technologies, and antibodies, etc. We have a global presence in the Nordics, US, East and West Coast, and in Europe, and distributors in the broader geographies. And we have clients in all areas and fields of research, and our products are validated on Illumina sequencers, all the various types of sequencers. So when we think about diseases at Pixelgen, we look at them as an inherent cell function. So when cell function goes wrong, it causes diseases. And these functions are executed by proteins, and not by proteins acting alone, but forming clusters and complexing together with other proteins. And this dynamic protein interactome is a vast and unstudied mechanistic dimension in translational research. And with translational research, we mean clinical research, where you're studying patient samples in cohort studies. So this has massive potential for novel types of biomarkers, new drug targets, and discoveries of completely new therapeutic avenues. And this dynamic interactome is what we now can enable to study at the single cell level with just a simple chemistry kit that can be run in any lab that we call the Proxiome kit. And it is based on this proximity network chemistry that we will soon describe to you. So when comparing proximity networks to other technologies for proteomic analysis of cell function, we have multiple well-known technologies like flow cytometry, mass cytometry, and CiteSeq, providing higher and higher multiplexing for single cell detection, but with no spatial resolution beyond the fact that the proteins are existing on one cell. So if we want to increase the spatial resolution and understand the protein organization on cells, we need to use a microscope typically. And microscopes can have 2 to 500 nanometer spatial resolution and have a 3 to 4-plex capacity of colors at each time in a two-dimensional image generating process. Or you have super resolution microscopy with even fewer multiplexing levels, but very high resolution. So what we have developed with proximity networks is a high multiplex, high resolution of 50 nanometers in a very simple chemistry-based assay, where the cells are kept in suspension throughout the assay, making the data naturally three-dimensional. And this is accomplished using only antibodies with DNA tags, no fluorophores, and no microscopes. All you need is a centrifuge and a thermocycler. And at the end of the reaction, you get a PCR product that is fed into a DNA sequencer. So looking at the differences of various components of single cells that you can study, you have, of course, genomics, the DNA, the building, the blueprint of these components that make up the cell. Transcriptomics is the RNA, of course, which guides the production of these components and sends a message to make proteins. And the proteins themselves are parts lists usually. If you study single cell proteomics, you get a parts list of you have this, and this protein and the amount you have. But what we want to enable is the large-scale protein interactomics studies, which will enable you to look at the components assembled into functional systems. And that is what we are delivering with protein interactomics as the next level of depth of information on the molecular basis of cell functions. So what you do in our assay with the PixelGen Proxiome kit is you take single cells or cell-cell complexes in a suspension, and you combine it with our kit. You get a PCR product at the end of the reaction. You feed that to a standard sequencer and high throughput short read sequencing. The FASTQ file that's then generated is fed into our Pixelator freeware open source software that then generates these images of cells and localizes each single protein at 50,000 spatial positions on each single cell or cell-cell complexes. So for example, this is a B cell binding to a T cell. So this is done at massively high throughput compared to what can be accomplished with a microscope, enabling you to study clinical samples and so on. And we'll provide you some examples of that. So let's see if we can get this video started to see how the technology works. So this is a cell illustrated with proteins on the surface of it. To it, we bind antibodies with DNA tags. Each tag has a unique molecular identifier and a protein identifier. These are first copied with a gap-fill padlock probe reaction and then amplified by rolling circle replication, which forms a local few copies of each DNA sequence. Then each sequence is copied together with every proximal sequence. So the UMIs from one antibody that's next gets united with the UMIs of the next antibody that it's close to, forming a sequence-based network all across this single cell. So let's see if we can get this to the next slide. All right. There. Oops. So input to the assay, you have cells that are in suspension. You PFA fix them to lock them spatially into place. Then you bind the antibodies with the DNA barcode. And then the padlock probes will hybridize and amplify the UMIs and the target protein identities. And the RCA products that are close to each other are then united, the UMIs from them, with a linker oligo, which then is just amplified with primers, making the next generation sequencing library preparation step. So let's assume that we have a cell where CD45, CD3, and CD8 are close to each other. This will then generate a reads list of CD3 being linked to CD45 with each unique molecular identifier 1 and molecular identifier 2. And as we generate multiple reads from each single cell, we can form this spatial network of protein interactions. And each single cell is identified as one cell, not by one barcode, but by the 40,000 unique molecular identifiers that are connected to each other. So the software figures out what's one cell and what's another without any single cell compartmentalization needed. So this is a graphic representation of a single cell, 155 proteins, all colored in one color each, and their spatial positions across this single cell. So it's a massive amount of information that is generated by each assay. And we get multiple data outputs from the proximity network assay. We get the abundance, of course, of all the 155 proteins across each single cell, which is the typical data output you would get with CiteSeq or flow cytometry. So this enables you to do classical annotation of various cell types. You also get the clustering. So if one protein is polarized or clustered with itself, you get a higher clustering score. And this is done by spatial statistics and using spatial statistics to mine the graph network that is representing each single cell and seeing if this protein is close to itself more often than predicted by random chance. And then this clustering score is increased. You also have a colocalization score, which works pairwise. So each target protein is pairwise detected if it's colocalized more than predicted by random chance with itself, with its partner, or if it is segregated, which generates a negative colocalization score. For example, when a cell is migrating, it forms a front and a back end, and you have segregation of these colocalization scores, which is a process that we very often see in our data. Or if it's randomly distributed, you would have a colocalization score around 0. So looking at the abundance of these 155 proteins, you will then be able to quantify how much you have of these traditional markers. And the panel of 155 proteins has been curated and developed together with our partners and collaborators to select them. And each antibody has been validated extensively with flow cytometry to bind its cognitive target protein. So this is a ready-to-go panel of 155 proteins, and it enables you then to identify the various cell types. So doing that with the single cell specificity can generate this type of traditional UMAPs of various cell types and identifying your memory B cells, intermediate B cells, NK cells, monocytes, and so on. So after this process has been done at the data analysis side, we can start analyzing the spatial organization of proteins and comparing different conditions. So for example, comparing the organization of the T cell surface proteins in healthy individuals versus a disease, or non-stimulated versus stimulated, and so on. So with protein interactomics, what do we really mean here? So this data set exemplifies what a B cell protein colocalization heat map or protein interaction map can look like. So we have in this B cell heat map CD19 interactions, and it is interacting with CD21, 72, 37, 82, 102, and CD81. and also with itself here. And these are well-known CD19 interaction partners, validated from the literature. So it is a lot of activity going on the surface of these cells as the cell executes various functions like migration, attachment to other cells, and getting ready to form immune synapses and so on. All of these protein organizations are massively important for cell function, and that is what we can study with proximity networks. So proximity networks can be applied to the studies of hematological malignancies for patient stratification biomarkers based on protein clustering, understanding the mechanisms of drug action and resistance, and also finding novel therapeutic targets. So a study we did together with Chris Mason at the Weill Cornell is trying to answer the question, what makes AML more aggressive in relapse? So AML is a disease, acute myeloid leukemia, that is often contracted by young children. And after the first-line treatment, the disease often comes back. And when it does come back, it is more aggressive often than it was in the first instance. So we analyzed bone marrow samples at the time of diagnosis and compared it to the time of relapse. And what we found was that CD25, the IL-2 receptor, was much higher in its clustering score at relapse compared to diagnosis. And if we plot that as a cell, a blast cell, then the yellow part here would then denote where the IL-2 receptor is. So this could give us new hypotheses about the aggressiveness of these tumors in recurrence. They are possibly then driven by IL-2 based proliferation, which is known to cluster the IL-2 receptor. This could lead potentially to new therapeutic approaches to this particular cancer patient. Another study we did together with Aaron Vinney at Columbia. He studies also AML. And here, hypomethylating agents can be very effective in treating these patients, but in many patients, they have no response. So what we built was a predictor based on abundance, polarity, and colocalization scores for a small pilot set of responders versus non-responders, resulting in 94% accuracy of prediction. So here you can combine the classical abundance-based measurements with our novel scores of polarity and colocalization to improve the prediction of drug response. So another study we did was just comparing cancer cell lines of Burkitt's lymphoma with healthy B cells. And these Burkitt's lymphoma cell lines are B cell-like, and they have a very different spatial organization of their surface proteins. So the Raji, Dowdy, and Ramos cell lines have an exclusion of CD40 from a colocalization domain containing CD20, which is present together on the healthy B cells. Also on these cancer cell lines, CD38 and CD37 is much closer to each other than on the healthy B cells, forming a potential bispecific drug target for the future. So this data was presented together with Illumina in a tech note that we developed together with them. So a lot of researchers are using Pixelgen in their research around CAR T therapies to differentiate molecular mechanisms of action by CAR Ts and understand more about the molecular basis of why one CAR T cell type works better than another, and so on. So in this case study here, we took a research-grade CAR T cell and bound it to Raji cells, and they then form cell-cell complexes. And we can identify cell-cell complexes in our data by finding the graph networks with highly polarized highly polarized B cell markers, which would be the cancer cell, and T cell markers, which is the CAR T cell. So here's one such component with the T cell part here, CD2, and CD40 on the other end here, highlighted. And over time, we can study these and fix the reaction over time. You start out with having 100% Raji cells or 100% CD2-based T cells. And in the co-culture, we form complexes. 20% of them are in complex at about 4 hours. 24 hours is 6%. And of course, these target cells of CD20 are killed over time. But where it gets really interesting is when we study the spatial organization of these cells during this process. So the CD19 CAR T cells bind to the tumor cell, and during the tumor cell killing, a patch of the dead part of the cancer cell is left on the CAR T cell in a process called trogocytosis. So this is a process difficult to study without a microscope, but we can do so at very, very high throughput, finding CAR T cells without patches, 1 patch, 2 patch, or 3 patches. And these patches then occur on the CD25 positive CAR T CD8 T cells. So we can then learn more about what are the molecular mechanisms of these CAR T cells, because the trogocytosis process then leads to blocking of the CAR Ts from binding the next cell and potentially also overstimulating them, leading to exhaustion. And each patch is comprised of multiple B cell markers, CD20, CD40, CD54, all together in one area on these single cells. So we've also done research into immuno-oncology and drug development. Here we can discover biomarkers of drug response based on protein clusters, and also discover novel mode of actions of therapies by their effect on cell surface protein dynamics. So here is organoid or a spheroid of breast cancer cells was combined with PBMC and the anti-HER2 antibody trastuzumab, also known as Herceptin. What happens here is that the same process occurs. NK cells from the PBMC co-culture bind to trastuzumab that is then bound to the breast cancer cell line in the spheroid organoid culture. And these patches of trogocytosis then also emerge on the NK cells, which are the mediators of these cell killings. And we can also study the colocalization matrix across multiple marker types in this experimental setup, where we find new insights into how NK cells actually perform the cell killing in the organoid co-culture. One other study in drug resistance development in hematological malignancies, I believe this was also in the AML area, was done at Uppsala University, where they studied the spatial organization of leukemia cells undergoing fludarabine treatment. So cells co-cultured with this treatment would then spatially reorganize cells over time, forming new insights into mechanisms driving chemotherapy resistance. So this technology here gave new clues as to why, over time, cancer cells develop resistance to fludarabine treatment. This is a type of study that we've done multiple times with other drugs as well. And it can give new clues and new therapeutic avenues how to combat these types of resistance developments. And of course, if cancer cells were not resistant or did never develop resistance to chemotherapy, we would be able to cure most patients right up front with the first-line chemotherapy. Here are some examples of publicly presented Pixelgen results all around the world in various disease research areas. Feel free also to reach out to us if you'd like more examples about application areas and what type of research questions that you have and how you think you would like to apply Pixelgen to your research. And we will help guide you with that. So the kit that we have launched on the market is the Pixelgen Proxiome kit for 155 protein markers for this sequencing-based protein interactomics technology. So each kit contains 8 reactions for 1,000 cells per reaction. And there's no capital equipment investment needed. So it's just a reagent kit and free software. Some people have called it like a sample prep for protein interactomics. It's very simple to use. You get a PCR product at the end that you feed your next-gen sequencing instrumentation. But I'll pause there for some questions and leave the floor to you guys. Thank you for listening.
Yeah, thank you, Simon, for the presentation. I've actually opened up, there's two sets of participants, panelists, and attendees. I've allowed everyone to have the ability to talk. So if you have any questions, feel free to come off mute and ask, or you can also use the chat box. We have an initial question here from Artie. Have you tested this in identifying virus host PPIs?
No, we have not. We don't know. Maybe one of our users have out there in the world, but not to my knowledge. It would be a really interesting study. It's been suggested a few times, and I think the spatial organization of immune cells that are infected with the virus should be very different. So that's definitely doable and a very interesting question.
Any other questions from attendees?
Hi, thank you. Great talk. Do you hear me?
Yes.
Yes, Leah,
Oh,
we can hear
great.
you.
Sorry. I was just wondering, um, is the protein markers selected customizable or is not yet something that you guys can do? If one marker
Yeah.
is if one marker of the 155 was missing or for a particular study, um, could it be added?
Not at this stage, but what we do is
Okay.
we would love you to let us know which proteins you would like to have in a panel. And over yearly, we will update the panel containing more and more of your favorite proteins.
Okay. Thank you.
Simon, we have a couple additional questions from the Q&A box. Are there current plans to expand the protein markers to other cell types or just immune cells?
Right. For now, it's going to be immune cells for quite some time going forward because it covers most research areas. The immune system is involved in every disease that I can think of at least. And also antibodies are readily available there. But please let us know which type of cells that you are interested in. We've worked with so far a lot of PBMC type samples, bone marrow samples, um, freshly dissociated solid tumor samples, looking at the immune infiltrates there, and multiple types of co-cultures, cell lines, and so on.
Yeah, thanks, Simon. I think you answered, but there was kind of a follow-up here. Other than blood cells or culture cells, will we have applications for human or animal tissue samples?
Not yet.
Thank you. Another question from Alejandro. Similar to organoid models, is there any experience on in situ tissue samples like solid tumor types?
The only experience we have there is where we've had users accessing fresh tumors and dissociating them into single cell suspensions and running our assay on that. But generating protein interactomics information from a complete piece of FFPE tissue is not currently available.
Simon, there is a couple other questions here. Joseph Olives has a question about bioinformatics. How bioinformatically compute intensive is the single cell network calling method with and without the cell morphology component and relative to current single cell calling by unique bead cell index methods of single cell seq? What is kit price? If you
Yeah.
want to address it first, I could do the second part, Simon.
Yes. So I think it's bioinformatically and computationally as intense as traditional single cell RNA sequencing and CiteSeq. So if you're used to that type of workflow and analyzing that type of data, this is very similar. And we have field bioinformatics scientists ready to train other bioinformaticians on this type of data analysis, or you can check out our tutorials and learn as you go. And that's another option.
Oh yeah,
And
thanks. I can field Joseph the
yes,
second part of your question regarding the kit price. So the kit price for the kit, as Simon stated, it is for 8 samples. The kit price is $14,990 US dollars. That's for 8 samples. There's also another option, which is a full service for 8 samples. That would be the samples would be sent to our headquarters in Stockholm to be processed, and that would be include the kit, reagents, labor, sequencing, and then also some first pass data processing and bioinformatics, which is about 40 hours or so of analysis. And the deliverable to your lab would be the rich data set that Simon had presented, including the metrics for abundance, clustering, co-localization, and 3D imaging. So that is actually 25,990 as for the full service. So the kit is 14,990 and the full service, which I described, is 25,990. Simon, we have an additional question from Leah. She says that she is curious to understand how cell-cell interactions can be measured. Does it come from fixing the samples first?
yes, that's absolutely correct. So the first step of the assay is a mild PFA fixation without cell permeabilization. And if two cells are then interacting, they will be locked in place. So what we have seen in multiple studies from PBMC, there are low levels of circulating B T cell complexes, platelets binding to monocytes, and so on, which is a rather difficult event to see with traditional flow cytometry. And being able to, for example, differentiate what is a cell-cell complex in CiteSeq or single-cell RNA sequencing compared to an actually two cells interacting is not possible. But with our graph network data, where the entire sprawling network encompasses two cells with highly polarized B cell, T cell markers, we can see those very clearly. And that comes from the fact that they're fixed first and then processed throughout the assay as fixed cells. And of course, our antibodies are then validated to bind the PFA fixed versions of all of these proteins.
Great, thank you, Simon. Yes, I'm looking at the chat box and the Q&A. I think we have addressed all of the questions. If there's any additional questions, um, yeah, feel free to send us. There is actually an email that anyone could use for general info if you want to send any additional questions or inquiries to hello@pixelgen.com. We will get back to you on that. Yeah, and I just wanted to thank everyone for joining today. And if you have any additional questions, feel free to reach out to us. We'd be happy to work with you all. So thanks again for joining, and have a great rest of your day.
Yeah, thank you for listening.
Thank you, Simon.